Research Director here at the Elling Eye Center. On behalf of the staff, I want to welcome everyone to our third lecture in our Florida Scholars Speaker Series. We developed this lecture series to highlight the academic research from our state in areas that were of scholarly and personal interest to Mr. Elling Eye. Those areas include the languages, literatures, and cultures of East Asia, as well as the diverse cultures of Central Asia and the Indian subcontinent. His passion for the vast history of this region is reflected in the extensive holdings of the center's library. The 72 acres on which this library stands reflects Elling's deep interest in horticulture, ecology, and the natural environment of Little Sarasota Bay and Florida and the Florida coast more generally. While Elling's fascination with the cultures of indigenous people of the American Southeast can be seen in his endowment of a position in the University of Florida's Department of Anthropology, dedicated to the study of the languages of Florida's native peoples. Today, we are very pleased to welcome Dr. Sandra Gilchrist, Professor of Biology and Marine Science at the New College of Florida and Chair of the Division of Natural Sciences. Dr. Gilchrist received her PhD from Florida State University. Her work focuses on the biology of invertebrates, including research on crabs and their interactions with other organisms. Her recent work has focused on the interactions between octopuses and hermit crabs in reef and seagrass environments. She has also studied mangrove tree crabs, which many of us are familiar with from kayaking around the mangroves here in the area, and hermit crabs. Dr. Gilchrist teaches a variety of courses on invertebrate zoology, mangrove ecology, and oceanography. She has received grants from the Sarasota Bay Estuary Program for K-12 teacher workshops and summer science programs for underserved middle school students to learn about the marine environment. She has also worked to expand the college's marine science program to underserved students with a gift from the AT&T Foundation. Her lecture today is titled, Often Overlooked But So Important, The Roles of Invertebrates in Shaping Marine Systems. Please help me extend a warm welcome to Dr. Gilchrist today.
I am apologizing for sitting. I have hurt my foot, and I'm afraid if I stand, I'm going to fall over. So please do excuse me for sitting during this talk.
Yeah.
And I do welcome all of you and thank you for coming. I'm used to having students ask me questions. So if you have a question during the talk, please raise your hand and I will try to recognize you. I do have some spots where there's an obvious break in what I'm presenting, so we can stop at those breaks and you could ask questions about those particular sets of slides or sets of ideas. So please, this will be rather casual in that sense. It'll be more like a learning experience for everybody than a really high formal lecture to you. I'm used to the interactive part of it, so please feel free to do that. I want to talk about the invertebrates because they kind of get a little bit overlooked. And the invertebrates, we eat them, no problems there, the shrimp, the crabs, the lobsters, and that sort of thing. But when you start to think about a system, we think about those large mega organisms like sharks and manatees and things like that. So I want to talk about these creatures and sort of introduce you to what I do in Sarasota Bay and abroad. So if I can get this, backwards. Can I go backwards? I'm already in trouble and it's only the second slide. Backwards.
There you go.
Okay. All right. So the invertebrates actually play a major role in all of our ecosystems, whether it's an underwater system like a seagrass system, or whether it's a mangrove or pine system, pine barren system. The invertebrates are the key. And so I want to focus some of your attention on that and ask you to see what you see in these images, and I'll attempt to use this without blinding you with the pointer. And most people, when they look at these images, you see this organism, okay, the sea cow or the manatee. but there's so much more out here in this seagrass bed. And when you look here, when I did a survey of my students and I asked them not to shout out what they see, they talked about fish. And when you look, there's so much more here than this lonely fish that's sitting here in the middle. So I wanted to try and talk to you today about some of the things that I do with some of the invertebrates. And I'll see if I could click one slide, not two or three. Maybe one slide. Yes. Okay. And the project that I'm going to introduce in Sarasota Bay is one that I've been working on for a few years now and is progressing into a more extensive project. And that is looking at microplastics. And some of you have probably heard of microplastics before. Some of us are wearing things that will shed microplastic fibers. If you're wearing anything polyester, any of the sports clothes, a lot of the shoes, things like that, you're going to shed some of these fibers in your laundry, or you're going to shed some of them just by walking by. So my interest in the microplastics is how do they get into our bay and what kinds of organisms are consuming them? And then what are the effects of that consumption? And I'll probably tell you some things that you're not really thinking about in terms of that, because the ultimate consumer is you, okay? And what we did was we worked in the area from basically Bowley's Creek down to Roberts Bay, and we were taking samples of plastics, taking samples that would tell us where these plastics were. And we used two types of sampling because what we noticed was that if you use one type of sampling, you got one answer. But if you use a second type of sampling, you got a different answer. So to make our study comparable to other people, we decided we were going to use both. And I work at an undergraduate institution, and so there are lots of undergraduates who are very happy to spend the day out on the bay on a boat and have an excuse to do so from their other classes. So in the guise of training, okay? And many of these students are actually senior students who are working on senior projects. And so getting a little bit away from those senior projects really is a boon for them because they can think about something other than what they're doing. So we sampled the surface water with what's called a manta net, and I will show you a picture of that in the next slide. And we also did the sediments with using a geological core so that we could take at least 10 to 20 centimeters of the bottom sediment. This kind of limited us though, because we could not, by our permit, sample within a grass bed, because you don't want to have the net perhaps or the boat disturbing the grass bed, and we certainly didn't. So we had to kind of sneak around the edges and try to get our samples from there. What we took were multiple tows at each of 5 stations, and we took multiple cores at each of those 5 stations. And we also took some water conditions like temperature, like what we call turbidity, which is how cloudy does the water look. pH, do we have any issues where we have high acidity in an area? And sometimes you do. You have high acidity from areas where there are mangroves. because those mangroves produce leaves which degrade, and that changes the pH of the area. So we wanted to kind of have some of those areas to know what kind of interaction pH might have with those microplastics. So it was a study where we were really trying to focus on a lot of different things. And again, I was fortunate that I had undergrad students because they all wanted to do something different. So some would take those water conditions and work with those and correlate those with storms and things like that. Others would take the cores, sample through the cores and see what was there. They were interested in the size of the grains, the kinds of plastics, that sort of thing. And then I would have yet others that wanted to do the manta net because that was the surface. And so there are lots of different things in that surface. And I want to say here, I'm very grateful to HDR, which is a national corporation, but one which we have a branch here for funding this research. We used the funding from HDR to use our boat and also to buy some of our basic equipment. See if I can get it. There it is. Okay. So I'll point out some of the different areas, and this is basically, it's very hard to see, but there's a core here, and I'll show you in the next slide that what the core actually looks like. But this is us deploying the core. This is deploying that manta net that's going to go along the surface, and you'll notice here there are floats, and that holds the net up to the surface. And this is the net itself before it's deployed. It has a little bucket on the end of it that catches everything that comes to the net. And what we find a lot of times are things like these. These are ctenophores or cone jellies. We have a lot of those in the bay during certain seasons. And primarily we see them on moss during the spring. And you feel like you can almost walk on it in the spring. And yes.
Well, I have read that, I wonder, I can't paddle in the bay frequently. When I saw that, I wondered if that was due to a deterioration in the water quality in Florida.
No, they come every spring. They usually are offshore, and during the spring, the prevailing winds will push them closer and closer to shore. They really can't propel themselves very well. The only way they can really change their direction is they can either stay high in the water column or they can sink. And when they do that, they can catch some different currents. So they'll move into shore. But the trick is when you get into shore, not to get onto shore. And so it's really a trick for them to be able to get in where the plankton is great and they can feed versus getting pushed onto shore where they're going to dry out and die. So really in April, you'll start to see these animals coming in. And the cool thing about them is they bioluminesce, which means that they create their own glow. And they glow in a range that's a little different from other organisms. They grow in glow in a bluish green, but also fade to red. And that means that it's going to a lower energy in the light that they're producing. So it's really kind of an interesting set of organisms, these cone jellies. But what we find is that in the cone jellies, they are ingesting microplastics. And it's easy to see in them because they're clear and their digestive system is clear. So you can really see it very, very easily. And this is what we look at as microfibers. It's not super jump out at you outstanding, but these fibers can accumulate in quite high numbers. And in organisms like these cone jellies, rather than defecating them out, they'll kind of push them to the side, inside the body. And so you'll start to get them packed in, and that can really affect their digestion over time. So we're interested in that. But down here is an example of what we see as microplastics. They kind of look like bluish sand grains, but we have the bluish, the reddish, the greenish, the yellowish, and the whitish. And once you start looking and finding these microplastics, they pop out at you. But a really interesting way that we've used is we use a black light, and the plastics glow. So you can easily tell the plastics from some of the natural background. And so it's easier to separate them from sand, if you will. So it's kind of a trick of the trade that we just found out by accident. Particles were separated, as were the fibers, using standard EPA methods. And what we found out is that in Sarasota Bay, more fibers than we did particles. And that can be a problem, because with fibers, if you're doing your laundry, if you're washing, whatever, these fibers are going to go out with your wash water unless you have a special filter. And most people don't. You don't know to ask, does this machine have this kind of filter? And even things like a car wash doesn't have these kinds of filters. So these fibers go right on through it. Some of the wastewater plants do have filters that stop a lot of the particles. So we are collecting some of those and keeping those from going in the bay.
Is that a retrofit you can do on the machine?
No and yes. If you have a newer machine, yes. If you have an older machine, like 5 or 6 years old, you're not going to be able to do it. Yes,
I have seen a product that you can attach to the wasteline.
you can do that. But many of the products you get will clog within a week. And so a lot of people, even though those products are there, they get disgusted with them after a time, because you're buying them and they're not cheap. And then the question becomes, what do you do with it?
Right, yeah.
What do you do with it once you get it collected? And I could tell you that what we would do is put it in acid and dissolve it. But most of you probably aren't going to be able to do that at home.
Mushroom farm.
Yes.
Can you clarify the microplastic? You're saying it's not the water bottle that falls apart, it's something else you're talking about.
Water bottles can be a part of the problem, but it's mostly things that are smaller particles that, for instance, you may have a lot of microparticles in salt that you don't think about. You may have microparticles that will rub off of something plastic in your house. You sit in a plastic chair or a chair that has high fiber content. That's on your clothes and it'll get washed off in your laundry. So there are lots of different places that we can get these plastic particles. And initially, they're kind of jagged and at angles. But as they're in the environment, just like stones, they'll start to round out in the environment. So you'll start to see them as beads rather than those angular particles. Yes?
If you mentioned about the particles getting washed into the bay and that, does a septic system stop them from going out the bay?
Unfortunately not. Many times the septic systems work fairly well, but during times of heavy rain, sometimes that'll flush the septic field and unclog it, which is good for you, but not so good for the bay. And there are also creeks that'll run off from neighborhoods, the tidal creeks. Sometimes after a really strong storm with a lot of rain, you can sample there and you don't even need a microscope to collect some of the fibers or the beads that are coming off. So in general, they're effective, but not all the time, okay? So let me see if I can get one. Okay. No. I got it. Okay, so this is a close up of the manta net in action. And you can see that because of the floats, it's skimming along the top. And our manta net, this is a really nice one. Ours is only half this size. Ours is a half-meter net. This is a meter net. But we had a half-meter net, and we would collect for 10 minutes. And inside, there is a current meter. And it will tell you how much water is flowing through, so you can calculate how much microplastic there is per amount of water flowing through. So it's an easy calculation to do. This is an example of the core. It's called a gravity core. And basically we can stand at the back of the boat. It's weighted, not the boat, but the core, and drop it over. And it will penetrate 20 centimeters. So it's a pretty heavy, it's not easy. That's why I had, in the previous slide, I had my boat captain who is in much better shape than me deploy the geological core. But this geological core has a piston on the end that helps to drive it into the ground, into the sediment. And then you put a cap on the top that then keeps the sediment and water from flowing out. It holds it from the top. And then you just put the core into a sample container and go back to the laboratory and sort through. So you can imagine we had hundreds of pounds of sediment to go through, and how do you really work with that? Plastic floats. So what we could do is put the sediment into a system and add a little bit of really important chemical to it. It's called Dawn dish detergent. Okay, and that would help those plastic bits to float. So we could skim them off the top and work fairly effectively with each of those samples.
What was the time that you did this, the time duration?
You mean time in terms of the toes or time period?
The time period of the study
Okay, it was about 14 months
from,
from 2019, the beginning of 2019 to 2020.
was there baseline data from before that?
No, and no, and yes. There was a baseline study that was done through Mote Marine Lab. And we used that baseline study to kind of think about where we wanted to put our sampling sites, but they didn't go as far in the north as we wanted to go, up to Bowley's Creek. They focused primarily in South Bay area. So we wanted to catch some of the marinas and some of the areas in the county that didn't have city septic, that had septic systems in the yard. So we wanted to catch a little bit different group than what that first study did. But what we found is that I would say reasonably for the sampling type, we had comparable results in the areas where we overlapped with their study. But there's also going to be change from year to year.
Sure.
And we found, of course, during the pandemic, when we were doing some sampling just around college, that some of the types of materials went down drastically, especially around the marinas. Can you guess why? People weren't there. They weren't taking out their boats. So it changes for a lot of different reasons. Yeah, a lot of reasons.
I'm wondering if, because the city put those bioswales in, if you were able, how they measure it.
most part, for things like fibers, they're not gonna work. For some of the bits of plastic, and especially for things like styrofoam, they will. But they have to be cleaned so that the bits don't grind down and go right through the soil and into the bag. But it's nice for it to catch the styrofoam. The styrofoam is one of the worst things that we find out there. And it's one of the things that the creatures consume. And it gets packed in their guts, and then they don't have a chance. So this is some of our results. And I could show you graph after graph after graph. Your eyes would glaze over, and I would still be here 4 hours from now talking about it. So I'm just going to do some summaries here so you kind of get an idea of what we found. Basically, we found that when you do toes, that is when you're at the surface, you get a different result than you do at a core. This is not surprising because toes are giving you particles that are floating. Cores are giving you particles that are already coming. Is organisms glom onto those pieces of plastic or styrofoam, and as they do, sink and become a part of the sediment. So we're not surprised that this happened, but it does make it interesting to try and compare to other studies. Studies that only did toes, what does that tell me? Can tell you what's happening now. Can't tell you necessarily what's happened over time. Cores can tell you more of what happened over time, because these are the ones that are deposited. They're in the bottom. They're things that when a worm goes through there, it's going to go and snuffle through that sediment. Or clam, when it settles down in there and kicks up the bottom, it can filter that material. So for us, both of these are important pieces of information. Cores, we got more beads. This is not surprising. Toes, we got more fibers. This again is not surprising. Fibers float. Takes them a while to get colonized. So these are not surprising results. So the question is, how can we use this information? Now that we have this background, now that we have an idea of the distribution of where certain areas are that may need more attention, what can we do? So we have a project now where we're looking at organisms that graze on seagrass. Things like these characters, which you can see better here, these are sea urchins. And some of you who eat sushi may have eaten sea urchin eggs. So what we're interested in is when the sea urchins graze on these seagrass blades, the seagrass blades particularly will glom those plastics onto the grass blades. There are lots of organisms that will catch them and hold them there. So what happens when these organisms eat it? Do they concentrate it or do they poop it out? And one of the places that we think they're concentrating it is in their eggs. And this is a picture of sea urchin eggs and sperm. So this is female, this is male. And these are what we call free spawners. They spew their eggs and sperm out into the water. When the female starts releasing her eggs, there's a pheromone that alerts the male, hey, there are eggs out here. The male starts spewing out sperm. And so they're free spawners. So we can look at these eggs in the lab before they're fertilized to see if it is in fact the eggs that may have fibers or beads or particles, or is it a combination of the egg and the sperm? We suspect it's the egg. It's bigger, got more room in there. Down here at the bottom, we're looking at a different kind of egg. And some of you may have seen this kind of egg. It looked kind of like spaghetti. If you walk in the intertidal, you may see it in the seagrass bed. Looks like green spaghetti about January. So they're kind of coming on. But this is a sea hare, and I promised you would see slugs today. This is a sea hare, and it packages its eggs into these spaghetti-like strings. But these eggs are already fertilized for the most part. But what they do is they lay a few eggs at the beginning that aren't fertilized. And then all the rest of them after that are. So we have both fertilized and unfertilized eggs that we can look at with these. Now, hopefully, I'm not sure how I can do this, but I'm hoping I can show you a little bit. Can you help me out?
What would you like to do?
I would like for a video. Like if it would
Oh, geez.
really high tech video.
Where is it?
It's up right there. You got it.
Got it. Hold on.
There it is. This is the ragged sea slug, and they're beautiful. They have been very important in neurobiology because they have what we call giant neurons. And so they've really been very important in the study of how neurons, like in your brain, function. But in our bay, these organisms are found primarily in the seagrass areas. They graze on the seagrass just like the urchins do. This one is in the laboratory. They're very easy to keep, which is a key anytime you're doing a study in the lab. And you can see it's got a personality. It's coming up now. This is its mouth. You can see its mouth opening and sort of scraping off the glass. And so we can accelerate the amount of plastic that it will consume. And we can enrich its environment to see if it really is packing some of that plastic into the eggs. Now, who cares about urchin eggs and sea hare eggs? We do, because there are lots of things that eat these. And those things that eat them ultimately are things like fish. And who eats fish? We do.
But we already know that those things are shown up in our tissues.
They're showing up in places you don't want to know about. Because some of the places they're showing up that some people will sample, not me, are human feces. You can dig through, and using those same EPA tests, have the plastics float, and you can collect microparticles from human feces. That has not been done in Sarasota or Tampa Bay area, but my suspicion is it's not far off. The other place that you're going to find it is in your table salt. And if you use some of the fancy stuff that's natural, it's higher than in some of the fake salt, like the potassium salts and things like that. So I do, in my teacher workshops, I do a study with the teachers and have them guess which of the salts are going to have more plastic and which are going to have less. And almost all of them say the ones that are more expensive have less. Exactly the opposite. Usually the ones that are more expensive have more. So you're getting them not only from the food you eat, but also from the condiments that you're using. And we're finding that out by looking at fecal material. You're ingesting and you are defecating some of that material. So I'll ask real quickly, do you have any questions on what I've already, yes?
Trying to understand what the harm is with these microplastics.
For you, for humans, you can get in your intestines Some of these particles collected in little sacs or pockets called diverticulites. And you can get diverticulitis because your intestines will try to enclose it like a pearl. And when that gets irritated, you'll get an inflammation that will cause you to have problems with diarrhea that could cause you ultimately to have to be hospitalized if it's severe. So it can cause that kind of a problem. We're not 100% sure with the fibers what's happening, but we find them, of course, coming out through the gut, but there is some indication that they do get into the kidneys. And there are sections that have been done and shown that some types of fibers are collecting in kidneys, and we really don't know what kind of effect that has. So we're still a work in progress with a lot of this. Did I answer your question?
Yeah. Are they, I don't know if I'm using this word the right way, are they soluble in a way where the chemicals of them like leach into your system at all, or is it more the issue of their kind of their physical presence? They can leach some things out depending on the kind of plastic, but for the most part, it's their physical presence.
I've read some places that they talk about plastics and it's hormonal effects on human beings. Is this part of it, or is this wrapping things in place?
There can be with certain kinds of plastics, leaching that does give hormonal problems, and we find areas where, for instance, with alligators, where there are some issues, both with birth control medication, but also with plastic that will leach out material from there. So it can be both. But again, really just studying what's going on in humans. We're the apex, we're the end product here. But we put it back in the system when we poop it out again. Yeah.
Do these concentrate through the food chain?
Yes, absolutely. Absolutely. Yes.
Did these lead to bleaching of the coral?
This absolutely does not, fortunately.
Okay.
Bleaching, and I'm going to talk about that in just a minute. Bleaching of the coral actually is more with temperature and with other kinds of factors, pH and temperature.
Okay, so not red tide either then?
Most of the places where we're talking about heavy bleaching, like in the tropics, no, it does not. So I'll go on then to the second part of this, and I'm going to talk about some corals. So I'm asking you to shift gears and move from microplastics to a system down in Honduras. I don't know if anybody has been to Honduras. It's a really interesting place. It's even more interesting now. Trying to get in, we go every summer with a group of students. And where we locate is this place right here. It's called Cayos Cochinos, the Hug Islands. And this is the area where we work. It's now called Eagle Ray Bay. It was called Plantation Beach Resort because it used to be a pineapple plantation on the island. And there was actually a landing strip there, and if you stand on top of the mountain, you can see it. But this is the kind of system we're working in, where we've got a sponge reef system. And I'm looking at my favorite animals, hermit crabs and octopus, and trying to find out how they shape this environment. And I claim, and I claimed in writing, that they are bioengineers. They manipulate their environment, they change it in such a way that other organisms take advantage of those changes. And I hope I'll convince you of that at the end of this talk. But the characters in the system are two different octopuses. And this octopus is not octopi. Like one of my pet peeves. It's a Greek ending, not Latin, okay? Octopus briareus. You can tell it because it has a dark ring around its eye. And that octopus is telling you, get away from me, when it does that dark eye. And this is our common one called Octopus vulgaris. We have vulgaris up here, so I can do some things up here and compare it to what I do in Honduras. And this is again our system. It's coral reef, but it also has quite a bit of sponge, quite a bit of soft coral, and these things called tunicates, which are one step below us. They have sort of a backbone, but not really, and they filter water. We have lots of them around here. These animals, these octopus, clear out dens in either the sand or something on the bottom or something up on the shore, cliff shore, okay? So they enlarge an area, but they also put stones in front of it. And they create these things called kitchen middens, which is basically their garbage dump.
Yeah.
Okay? And those dumps provide a lot of things for a lot of other organisms. Okay? So if you think about what can I get from a garbage dump? Well, you could recycle plastics. You could collect cans that people throw out that they don't recycle. You could tell by doing an archaeological dig in their middens. And the shell middens that are on this property are a result of people eating shellfish. So we could tell the percentage of their diet that was clam, that was oyster. You might find a carapace of a shrimp, that sort of thing. These middens have the same kind of thing, leftovers that they throw out. Okay. And our other players are the hermits. I won't go through all of the names. There won't be a quiz afterwards for this class. But you can see there are different kinds of hermits. Some brightly colored, some not so brightly colored. They occur on sponges, on the reef itself. This one, Pagoristes grayi, is one of the larger ones. And if you were looking at its claw, the claw would be about as big as my thumb, the end of my thumb. Okay, so they're not huge, they're not small either. And some of the predators of the octopus and of the crabs are these organisms called eels, the bane of my existence. Because just as soon as you get a site set up, an eel moves in because you've had activity in the area. So you have to be smarter than the average eel to do this, which isn't hard, they're not bright. Boxfish, which eat mostly the hermit crabs and small snails, and these coral crabs, carpilids. So they're predators, the octopus are predators, and they're prey. Most of the prey are hermits. So what do you see when you go to a reef? You're looking for octopus, right? Where's the octopus in this slide right here? This is it, yeah, right there. Yeah. You can see its eyes. Okay. It's kind of hard to find because they blend in and then it'll pop up and then back down, pop up and then down. This is what you oftentimes see, and you have to believe me, there are eyes in here, but it's sort of like the shadow. You know, it puts its arm across the front of it to block the opening to some extent, and then it sort of peeks out over the arm. But this is what you'll see most of the time if you know what you're looking for. And at night, you can see them much plainer, much clearer. This is one that's doing what we call a cloud over web. And it's basically putting its whole web and arms over one of these coral colonies. And then it takes an arm and pokes. And whatever jumps out, it catches. And it'll catch up under each arm some kind of creature. A fish, most often. And it'll eat the fish there. But if it catches something like a crab or a snail, it'll tuck it up under there and take it back to the den to eat, because it's gonna take longer to eat. So it's gonna forage over a certain area and then it's gonna take it back to the den. So how do we find octopus? Easily. Oh, no, don't move. We look, this is the octopus. This is its kitchen menu. We don't look for the octopus, we look for its garbage pile. And even though you can find the garbage pile where the animals already moved to another den, 9 times out of 10, you're gonna find something of value where these movements occur. And here's a picture of this organism a little closer up, and you can see it's in that, shadow nose mode with the arm across. But there's a hermit crab, and the octopus have a front door and a back door, almost always to their dens. Why do you think that is?
It's for that.
You can escape. If an eel comes to the front, you go out back. So most of them will have that escape. What we do is we look at this kind of material as a prey. And we've cataloged the kinds of prey that each different kind of octopus, in size of octopus and location of octopus, eats. In our initial hypothesis, where they would eat whatever's close by. I mean, that makes sense, right? Do you do that when you go to the grocery store? Sometimes, but most of the time you're choosy. And you pick your favorite thing. Or you pick the thing that's easy to do. If you're cooking it or eating it. They do the same thing. They will choose different things depending on where they are and what's abundant, but also what they specialize on. Some will be crab specialists, some will be fish specialists, some will be clam specialists. And so you'll find in these bins a lot of different things. This one actually had a sea urchin. I don't know what it was doing with that. Sea urchins aren't eaten very often by octopus, but it certainly had it in there. What happens is out here in these middens, these kinds of organisms find them and set up shop. Because hermit crabs and other creatures will come to those middens to find shells and to find food. Because octopus almost always leave a little bit left over. And so they just throw out a shell and it still has a little muscle on it. So you can go in and eat it. So things like lobsters and these mantis shrimp go in there and have a nice time of collecting hermit crabs and small crabs and snails that are attracted to these kitchen middens.
Did the octopus, see that one shell with a hole in it?
Yes.
Sea urchin hole with a hole?
Yeah, this one.
Do they make, somebody told me they actually make a hole.
Depends. They have a beak, which is very hard. And either they will crunch into it and kind of slurp out the inside, or they'll chisel around the edge. And so you can almost always tell when it's been eaten by a crab and when it's been eaten by an octopus, or when it's been eaten by a mantis shrimp. Okay. Yeah.
How deep is this water?
This is probably about 1.5 meters.
Oh, nice.
So shallow, yeah. You can just fall over it. Yeah. It's great.
Are octopuses endangered?
No. At least the ones we work on are not. There are certain ones that are in Asia that are, but octopuses are commercially harvested and they're commercially grown in mariculture. So people put out pots and attract the octopus, groom the octopus, and then bring them up and sell them at the market. But there are some smaller species that are a little rarer that are probably threatened or endangered. Now I can't get it. Okay, so we created our own middens. This is again your hermit, or rather your octopus in here. And your mantis shrimp, 2 different types that move into the area. And hermit crab takes a shell from the midden. This is the artificial midden that we created. We've done thousands of shells, hundreds of thousands. And what we're trying to find out, answer. Wait. This thing doesn't want to do it. Okay, thank you. What we find is that, yes, these are important middens for the hermit crabs, but not only for the marine hermits, but also for the ones on land. You've probably seen the little land hermits, the seen of Eide, in stores and things like that. People sell them for pets. But yeah, I'm not so sure. But this is what you see, land hermit. But what you find on land a lot of times, because shells are kind of scarce on land, there aren't that many snails that live on land on some of these islands. So from human, okay? And you see them in bottles. You do see them in plastic, unfortunately. But you see them in almost anything they can stick their abdomen in and clutch, hold on to. These are bad. But we can also simulate what happens if there's a storm. Some of these shells will wash up from the reef, and then the hermits will get it. But what we found is that, yes, indeed, that happens, and shell exchange does occur. This is an old shell, and this is one walking off in a new shell. One of the major transporters of birds. And what they do is in the intertidal, they will grab a hermit crab, peck out the inside, and then toss away the shell. So the shell is then high in the intertidal. But how did it get there? Because the octopus threw out the midden, and where the octopus was high in the intertidal. So they brought the shells closer to the land hermits, and then the birds kind of finished the cycle. So it's a really interesting system that we've been working on. So after many, many years, and in this particular spot, 18 years, we've learned a lot of things about this complex system. If you tweak a part of it, it's very resilient. It'll compensate over time. But what we've also found is that there are long-term cycles of population changes for the hermit crabs, for the octopus, and for the predators. And they don't all peak at the same time, which makes sense. They peak at different times and at different levels. So if we're trying to help manage this really important reef, this is in a bioreserve area, will it harm the reef to take a few fish? Answer is no. And especially if they're lion fish, get them out of there. Will it hurt for some of the artisanal fishermen to take some of the mantis shrimp and use it as bait? Depends on the amount. How much harm will harvesting the octopus do? We think that would be the worst thing, because they affect so many other creatures in the system. So we're advising the Honduran government to look at this as a system, not to look at it as individual organisms, and not even to look at the reef as a reef. It's a system, and there are many players that really help to shape that system, including humans. Let's last. They were very. This is the most. Pardon me?
Panama?
Yeah. I spent my postdoc in Panama at Smithsonian,
Very nice.
so I probably got about 300 moles.
I know it. I worked there for 2 years.
Oh, yeah? Yes.
Have you studied the microplastics in Honduras?
Yes and no. We just really started doing that. Unfortunately, what we're finding is more macroplastic. Like people have flip-flops that get blown over on from a boat, or Crocs, the shoes, Crocs that get blown off of a boat or fall off the boat or whatever, cans. A lot of the Hondurans where we work don't swim. So they use empty Clorox bottles as floats. And sometimes those floats get away. So we're worrying about macroplastic and things like diapers, where they put diapers, you know, now that you have these disposable diapers, they used to wash them out and hang them out in those great. If you live out on an island, you burn them, or nobody's looking, so you stick them out on the reef, because the reef's big. Oh, yeah.
Yeah. We're.
All of the polymers that are in the diapers are huge. So, yeah, it's, we haven't really done a whole lot of the micros, except in the sand, in the sediment. We haven't done any toes. Yeah.
Adult diapers are real, adult disposable underwear is real popular now too.
Well, in the islands, not so much.
Okay.
Mostly what you see are the baby diapers.
Here in Florida.
Yeah, unfortunately.
Yeah.
You see them even buried out on the beach, which is, you know, primary human sewage out on the beach. Yeah.
So, this has been going on, your research for 18 years. What do you hope to do with the results?
Well, I've published several papers on it. And like I say, our ultimate goal is to help the Honduran government manage this bioreserve. It's a beautiful reef area. It hasn't had very much disease, unlike part of the other tropics. And they're getting a lot of pressure when each of the governments change to open it up to more tourism, to open it up to more fishing and things like that. So we're trying to kind of help them in a way that is not, no hands on it, because that's not going to work. It's limited and rotational in certain areas, certain times of the year, and that sort of thing. So we're hoping to do that and to add to our understanding of the reef systems, because we can apply some of that to areas like the Great Barrier Reef. I've worked in Tahiti and other places like that to make comparisons of what are some of the overarching things that help shape these kinds of systems. And how far can you push it until the system changes? Till it changes from a coral system to primarily sponge reef. And those are some questions we don't know about, because they've had changes, but over hundreds of years. So we're trying to ask what's going to happen if we push it to a short-term frame.
When you go to these really remote areas, like Tahiti and Fiji, are you seeing a difference in what you're finding versus your college type of?
Yes and no. Some of the overarching principles are still the same. We look at some of the major predators, some of the predators, especially the invertebrates, the octopus, that are shaping some of those systems. And we try to overlay what we know from one system to the other on it. But there are so many differences, like changes in pH, changes in sea level, changes in just water, just water. And unfortunately, changing government, which can really be disastrous, because that can result in a whole different set of rules and open up some of these systems to a lot of damage very quickly.
What's your opinion on, I've heard of and read a lot about remedial measures like, trying to plant oyster beds and regrow coral and all this kind of stuff, and I kind of feel like, don't increase the water quality, how is any of that gonna be helpful?
Well, I have different opinions. And part of my hat is I'm also a population geneticist. And so it really makes a difference when you're doing some of these coral growback or oyster growback, whether they're enhanced, shall we say modified, where they're more resistant to disease or to drying or to differences in pH and things like that. I'm sorry, but I'm a proponent of genetically modified systems like that.
We have GMO.
Yeah, we do. I'm a proponent of that kind of thing. If you're gonna invest, don't put the same kind of organism back in there because it's only gonna die. So I have some different opinions about it, but things like oysters and some of those tunicates that I talked about, they're gonna filter the water.
Yeah.
The tunicates are, I believe it's about per hour that they filter. And so some organisms that are kind of not your charismatic megafauna, they're just kind of look like little dirt balls. They're going to help a lot, and so do the sponges. So I think we really have to think about this as a system. Think about what our final goal is. Do we want to treat it as a zoo? Putting things out there that we're going to replace regularly, or do we really want to work on the system? And I think, this place is beautiful in terms of the botanicals here. The ecology is going to be fascinating when people really start thinking about it. Yeah.
There's a program on Netflix, the Octopus and Me.
The octopus teacher. Octopus teacher.
Yeah.
Can you comment on it? How valid is it? Is it likely? Is
There are many things we can learn from octopus that they can teach us. We have had them in my laboratory before. They train us. When they want to be fed, if they don't want to be fed, they go off and it's like, okay, well, I'm not coming this time tomorrow. They train us very well. So I think there's some validity to it. I don't necessarily like some of the what I would say the over-interpretation of the interactions. I think that can be dangerous.
Okay, are you thinking of Sky Montgomery, Soul of the Octopus?
Yeah. Did everybody do the little flip page down at the bottom? It's a flip book. The bottom of that
Oh, I had
has an
that.
octopus moving if you flip the bottom pages.
Okay,
So it's fun. I think the first half of that book is reasonable. Second half, I could have torn it off and done.
okay, for your opinion, thank you.
Yeah, you're intuiting and anthropomorphizing an octopus. They just aren't put together the way you are. So that's my opinion. It's valuable in that it gets people to read and think about them, but it's not so valuable in people thinking that we know all about them. We really don't. Thank you so much for coming.